And next, we will be inviting Alex back to the virtual stage to present the analysis tools. So Alex, welcome back.
Yes, this is the presentation regarding analysis tools. And as we were discussing earlier this morning, the analysis resource of the IEDB hosts a variety of different tools, and some are predictive tools, and Bjorn was alluding to those, and to predict class 1 or class 2 binding, or immunogenicity, or processing, and so forth. I'm going to discuss with you briefly a series of tools that instead are, we call analysis tools, in the sense that they are meant to analyze data sets that the user might have generated or extracted from various sources, including the IEDB itself. But you're not predicting, per se, new epitopes or things like that. So there is a sub-page here of the analysis tools, and really the analysis tools are here under this tab, addressing a broad number of different applications, and these are really have originated from our own research or the research of other groups that had pointed out, oh, I wish I had a tool that would allow me to calculate this or that. You see that some of the analysis tools have a green lab flask associated with, and that is meant to indicate that these are tools that are accessible on the analysis resource, but they have not been validated as stringently as the ones that do not have the flask here. And this is noted on the bottom. The reason why, you might ask, well, if a tool is not completely validated, why do you make it available in the first place? Well, first of all, because we wanted the tool to be available as long as people understood that that tool may need a little bit more validation. And the other thing is that is useful for us in the sense that as the people utilize the tool, we receive feedback, and we have an opportunity to improve the performance or the usability of the tool. So, for example, population coverage is a tool that calculates the percent of a given human population that is expected to recognize or react to a particular epitope or sets of epitopes. And obviously that needs to be data that is available from the user, in the sense, if you have a sequence, you need to know that this sequence is, for example, binding to HLA A2.1 or immunogenic in the context of HLA DR beta 101 or whatever. And so basically what you do, you select, you input your set of epitopes with the associated restrictions, and then the tool will look up the frequency and calculate population coverage by the Hardy-Weinberg equations and so forth for that set of epitopes. And here you can see, you can select, for example, different locations, because as we all know, HLA frequency varies as a function of a different different ethnicity. And the output here is in a table form where it says what fraction of the population is expected to bind at least one of the epitope set, or how how many, say that you have entered a set of 10 epitopes, how many on average, epitopes does the average person recognize? And this is also shown here on this kind of plot that shows what percent of individual will bind 1, 0, 1, or in this particular case, or about, I believe, 30 epitopes were entered. Now, one issue is that the original tool extracted HLA frequencies from the resource allele frequencies.net, which is based on the original indigenous people in that particular geographical location. And so if I can make an example related to Italians, since I'm Italian and my grandfather was from Sardinia, and Sardinians have a very specific genetic origin and particular HLA types. So that what you would see in allelefrequencies.net. But the reality is that by now, the original indigenous Sardinian population is still there, but there's been lots of immigration, and the genetic makeup of the people that actually live in Sardinia is somewhat different from the original population. So to overcome this challenge, we also updated the coverage population tool by take advantage of the data from the National Marrow Donor Program. And so this will give you the snapshot of everybody living in the US, representing the mixture of different ethnicities you see here that are today captured by the Marrow Donor Program, and thereby represent really more of the who is actually living in the United States, as opposed to necessarily a previous indigenous population. And we will have both options available in the analysis resource. Another tool is the epitope conservation analysis tool. And this is a tool that is very useful to determine for a set of particular epitopes that you might be interested, how well conserved or not they are in a new set of sequences. So, for example, you have heard from Alba how relevant this is in the context of SARS-CoV-2 with a new variants coming up. You may say, okay, I have this set of epitopes. Are they conserved in the Delta, Omicron, and new Omicron variants, and so forth? And so what you do, you enter your set of epitopes here, and then you enter the protein sequences that you are interested in. And so for each epitope, you can have a result. Okay, this particular sequence that I entered is conserved, 100% conserved in 80% of the 10 sequences that I entered. And you can adjust the sequence identity threshold. So you can say, I want to know this particular epitope, what fraction of its query sequences that I'm submitting, in which fraction is conserved at least 70% or more. Another tool is the epitope cluster analysis. And the rationale for this tool comes from the fact that often you will have data from a set of epitopes or sequences which are largely overlapping, but not identical. And this is common because, for example, different studies or different labs may use different, slightly different sets of overlapping peptides and so forth. And then the issue comes of how can I cluster the data or the sequences from the different epitopes to make a more of an aggregate call. And there are different methods that can be used to do that. And the tool will explain in more detail the different options. But one issue is that if you cluster on the basis of epitopes having some degree of similarity with each other, you could generate a cluster that is very, in which the different members of a cluster are not necessarily all related to each other. So to explain this, if you make a set of overlapping 15mers, overlapping by 10, you would say, well, the first epitope and the second epitope share 10 amino acids, so they are clustered together. But so would be the second peptide and the third peptide. So you will generate eventually a cluster where the first peptide and the last peptide have absolutely zero sequence similarity. That is okay in some applications, but in other applications, you might want to have a situation where the all members of a given cluster have, say, 70% homology to every single one of the peptides in that cluster. And that is useful, but that has also the problem that the same peptide can be then found multiple times in multiple clusters. So that kind of inflates the number of epitopes, and also can lead to ambiguous assignments. And so here you see the tool, you enter your sequence, and then you can specify the type of cluster. And also we have generated a new updated clustering tool that will be discussed in more detail tomorrow. The restrictor analysis tools is another tool that utilizes information that's already available regarding sets of epitopes and that are being recognized or not in a human population that is has known HLA type. So, for example, what you have here in this example, you have a set of different peptides, and say, let's look at the first peptide here, this GINTI. This was tested in 50 different donors. And some of those donors responded to that peptide, and other donors did not. And so you can ask for each of the different HLA types expressed in the 50 donors that were tested, whether the donors that respond are enriched in particular expression of particular HLA type. And this is somewhat analogous of what is done in the case of odds ratio relative frequency of HLA, when you say, for example, that ankylosing spondylitis is associated with HLA B27. What that means is that people that have ankylosing spondylitis are more likely than the general population to express HLA B27, but of course doesn't necessarily mean that every single individual that has HLA B27 will have ankylosing spondylitis. And so in this particular case, A plus R plus means that these individuals have expressed that particular HLA-DR beta 30202 allele and also are responders. So there are 6 people that respond that express that allele. And when you look at people that respond but do not express that allele, none of them. So there are 0 individuals that respond that do not express this. Conversely, there are 18 people that have the allele that do not respond. So 6 respond and 18 don't respond. So about a third of the people that have this allele respond. And there are 26 individuals that do not express and do not respond. And so based on this, you can calculate the relative frequency and the odds ratio and the p-value associated with that event. So whether that is likely to be occurring by instance or is significant association. So this tool generates lists of potential HLA restriction that can then be addressed by further experimentation, or for example, make tetramers and things like that. Another tool that we have developed is the deimmunization tool. And the context for this tool comes from the fact that protein drugs are increasingly utilized either as monoclonals or as just proteins that are administered to patients. Actually been remarkably successful, but in a number of instances, there is an undesired event, which is the protein drug is recognized as an antigen, and there is an immune response that develops against the antigen. And so that can lead to loss of efficacy of the drug or safety problems. So what this does is scan a sequence of a protein that is submitted and runs a prediction for what are the most predicted dominant epitopes that might be contained in this sequence. And then for the specific dominant predicted epitopes, generates all the possible substitutions, and the computer program then checks whether the potential substitutions would be expected to decrease binding capacity of that analog substitution, and also check that by serendipitously, by mistake, you have not decreased the binding affinity for the epitope, but created a new epitope in a neighboring sequence to the epitope that was de-immunized. So another tool that is still in the developing phase is this PepSySco. PepSySco, I can never pronounce it correctly. And what this does is, looking at the sequence, predict how easy or not that peptide will be to synthesize. In over years, there's been a lot of data that has been generated, and there are problematic peptide sequences that can be flagged. And so this is quite useful when one makes sets of peptides for experimental testing to flag which peptides maybe needs to be synthesized or probably purified and so forth. Finally, there are tools like the expression annotations. This is very important, and I think we're very excited about this tool, because there's really been growing evidence that apart from the capacity of the peptide sequence to bind a particular HLA, another important factor in determining immunogenicity is how much of that protein or peptide is actually present. So that stands to reason that a peptide that is expressed in very large amounts will have a better chance of being immunogenic at the same level of HLA binding capacity of a peptide that is expressed in very vanishing amounts. And so this allows to enter the peptide sequence and then finds which particularly data sets is already available in the scientific literature where the expression level of the peptide has been determined by either RNA expression or other methodologies, and then returns the expression, relative expression level. And so this is a recap of the tools here. The population coverage with analyzed T-cell epitopes with no HLA restriction for what coverage a particular set of epitopes will give in one or more different HLA ethnic or geographical locations. A conservancy allows to determine whether or not or how much an epitope or a set of epitope is conserved in a target set of sequences. The cluster tool allows to cluster epitope based on their homology. The rate uses HLA frequency and response rates and HLA of the donors that are known to respond or not to a peptide to potentially infer the HLA restrictions. Deimmunization tool is a tool that is looking at protein sequences to not only identify likely dominant epitopes, but also identify substitutions that would be expected to decrease the immunogenicity of this dominant epitope. And the two most recent tools look at the capacity or the predicted capacity of a peptide sequence to be synthesized without issues. And the last, it identifies the peptides can be linked to expression levels as a useful tool in predicting immunogenicity. And with that, I'll stop the sharing.
Excellent. Thank you so much, Alex. We're running about 10 minutes ahead of schedule, so that means more question time and potentially wrapping up early too. But while I give Alex and Alba a chance to review the Q&A, just a quick reminder to all, if you did pop a question into the chat, please just copy it over to the Q&A. That's what our presenters are looking at to delegate out who will answer what. So much appreciated for that. And a couple of other things. We've given you a taste of the tools today. So we talked a high level about the analysis resource this morning with Bjorn, and this afternoon Alex has done a deep dive into the analysis tools. Tomorrow we'll be going into further detail. So all day tomorrow will be about the tools. So definitely join us for that. And now...
So I'll jump in and answer the question from Pangai Gupta. And the question is, can we identify T-cell epitopes in variable region of antibody? And the question is yes, but also the issue is there are additional considerations that can be kept in mind. So in general, I suspect that the question may be related of the deimmunization or the fact that sometimes using antibodies, even though they may be fully humanized, you still get immunogenicity that is directed against the variable region, because the variable region of the drug is maybe not found, or certainly not found abundantly in the general, in the normal situation. You come in and give a large amount of an antibody with a variable region, which is essentially non-self. So why am I saying that? Because when you look at protein immunogenicity and the likelihood that the protein will be immunogenic, one of the factors is the HLA binding capacity and how many different HLAs can perhaps bind particular fragments or region of the protein. But the other thing is whether the protein is self or not, because a self protein or regions of a protein that are self have a much lower intrinsic immunogenicity, because a lot of the T cells that could recognize that that protein or that region have been eliminated by peripheral or central tolerance. So the tools in the IEDB that predict immunogenicity should be used with the notion that also if a sequence is self versus non-self, it's likely to have a deep impact on immunogenicity. Alba.
Yes, so I'm gonna go with the first question, and I apologize if I'm butchering name or last name, Manikanta Bondara. Instead of doing all the steps, can we pass protein sequencing resource analysis and select TB epitopes, right? So if you are referring to my original query where I was showing all the filtering and so forth, that could be an alternative approach. The reason why I didn't do that is because, and I didn't have time to delve more into that, I was interested in things that were 100% conserved, but also the ones that weren't. And so that's why I did the analysis pipeline differently. But I cannot show with IEDB, you can use different ways to reach the same goals. So that's definitely one. Alex, you want to go with the next question, and I can take my second one after?
Okay, I will answer the question from Osman Yogurtsu that says, can the IEDB or analysis tool tell us how many T cells out of the total T cell population in a host are specific for a particular epitope or recognized as a specific epitope? And the answer is no. The reason why that is that is context dependent and donor dependent. So in order for a peptide to be immunogenic, it has to bind HLA. But beyond binding HLA, there are a lot of different things. And specifically, the size of a T cell repertoire is what you're referring to, of how many T cells can recognize that epitope. Well, two examples of main factors that can influence that is if that particular epitope binds with great affinity to HLA-A2, but not HLA-A3, the fraction of the T cells that recognize that epitope will be very different if the donor is HLA-A2 positive versus HLA-3 positive. So that is the genetic makeup of the host has a profound influence. The other point goes back to what we were discussing earlier. Is that sequence self-sequence or not self-sequence? That will have a profound impact. And likewise, is that a sequence, for example, that is are we talking about an acute infection or a chronic infection, and so forth? So in general, the fraction of T cells that respond is very context dependent. Back to you, Alba.
Thank you, Alex. Kamar Ayab, do you mean that the escape mutated epitopes could be expressed by HLA? So apologies if I went too fast into that, but essentially the point that I was trying to make is that, as we know, when T cells essentially recognize, there are multiple steps that happens in the process. So there is the processing component of things, and probably Bjorn is gonna delve more into that, but assuming that the protein is processed and the epitope bound to the HLA, then it gets expressed on the surface, and then T cells may or may not recognize. And so what I show you up to now is the capability of the peptide to still bound to the HLA molecules. And of course, our prediction did not take into account the capability of the TCR to recognize the peptide HLA complex. So the point that I was trying to make in my study is that the peptides that are mutated, they still retain the capability potentially to be presented to the immune system, and then it's up to the TCR to decide what it wants to do about it. Hope that answered your question. Back to you, Alex.
Okay. So Osman has another question. It is unrelated to Venali's tool, but you have time for it. Occasionally, scientific articles get retracted for various reasons. If this occurs with data related to an epitope, is the update reflecting the IEDB database? And the answer is yes, provided that that is flagged, in the sense that what usually happens if a paper is retracted or there is an erratum or a correction, the PubMed comes up with a record that says erratum or correction, and flags that that particular PubMed article has been corrected or abridged and so forth. And so we pick that up, and then we look at it, and if the correction is that, I don't know, the second author was misspelled, that obviously has nothing to do with the epitope data. But if a correction is that, oh, table 2 was wrong, and now this is the new corrected table 2, then yes, we do go and correct that that information. Alba.
Okay, I can answer, and probably Alex can help on that as well. For the epitope cluster analysis, is there any recommendation which minimum sequence identity threshold to use, or any tips on how to choose one? And so that's an interesting question. At the best of my knowledge, we didn't look at that comprehensively, but actually Alex and I have been working consistently in trying to understand a sequence identity that was that could be used if you look at CD4 epitopes versus CD8. What we've shown in different viral system, including coronaviruses, is that a sequence identity of 67% or 70%, if you wish, should still allow capability to cross-recognize the different epitopes, while for the CD8 we have been using a more stringent cutoff of 80%. And I don't know if Alex wants to add more into that, but that's
yeah. So that is totally totally correct for one of the application of a clustering tools. In other applications, you may be faced with a set of different sequences that have been reported as being immunogenic from 20 different papers and so forth. And you may be trying to answer the simple question, okay, is this essentially the same epitope? I mean, how many epitopes are in here? And then what you want to do is to say, okay, this really, there are 100 sequences, but there are really 3 epitopes here, because you can cluster all these sequences in 3 non-overlapping groups that are likely the 3 epitopes. And in doing that, also obviously depends on how similar they are in terms of are they just the same sequence but in different frame shifts, or is there a difference in the sequence? And so that it's a little bit of trial and error in terms of what sequence homology. And then there was another a question from Hor Alshani that was to clustering tools and say why clustering on epitope sequencing is not recommended. No, it is. But the point is the particular method you want to use depends on fundamentally whether you want each sequence to be found only in one cluster, or you can allow the same sequence to be found in more than one cluster. That is a fundamental difference depending on what application so you're doing, and then that's what the tool will give you some flexibility to do. And Nina, you were asking one of the answering one of the questions.
Yes, there were a couple of questions, and Bjorn kindly responded to one of them via typing, but I pulled up the tools which will hopefully consolidate the responses. But one of the questions was, does PepX leverage protein expression info from HPA? And Bjorn did respond saying, yes, the HPA one is possible. And the way you can find that, this is what PepX looks like. Select data source. Under this drop down, you will see the different options, and HPA is there. So thanks, Bjorn, for answering that. Hopefully that visual helps. And then the second question was from Annette. For the PepSySco score, what is the best cutoff for a likely success at synthesis? Again, this is just the PepSySco tool. And then if you go to the help tab, and all of the existing tools in the analysis resource do have the help tab, which I highly encourage you to view before using the tools. It does give you a step-by-step breakdown. But what it does say in interpreting the output, the score represents peptide synthesis from 0 to 1, with a higher score indicating more likely success at synthesis. So in short, just the higher the better, the closer to 1, the better. Hopefully that covers it.
So I'm gonna jump in and answer a question from an anonymous attendee, but asking, is it possible for CD4 and CD8 T cell to share the same T cell receptor that identifies the same epitope? And the answer is no. In general, that is not the case. So it is true what Alba pointed out, and in our analysis, when we look at what is recognized by CD4 and CD8, that sometimes you can find some hotspot that is recognized by both CD4 and CD8. But that means that peptides from that region bind to both class 1 and class 2 and are recognized by T cells. But in all likelihood, the peptides that will bind to class 1 and class 2 are different, and the TCR that recognize those peptides are also going to be different. It's just an overlap between regions that are recognized by both class 1 and class 2, or both CD4 and CD8. So then there's one question from Manikanta Bondara, which says, will membrane proteins, epitopes, have advantage than cytotoxic proteins in epitope-based vaccines? So let me maybe rephrase a little bit. The question is whether the topology of expression of proteins is important in determining immunogenicity. That is certainly true. In fact, some of the a success story of reverse vaccinology was the development of the meningococcus vaccine. And what was done there was actually to, once with the full genome of meningococcus became available, scientists that developed that vaccine looked at what proteins were expressed, likely to be expressed on the surface of a bacteria, and likely then be recognized by potentially by antibodies, and then they screened those proteins and identified 3 proteins that were particularly immunogenic. And that is important because things that are expressed on the surface and are recognized by antibodies are also selectively more likely be recognized by CD4 T cells. Now, CD8 T cells, it's a different story because there it's not particularly important to be on the cell surface. It's important to be made and degraded and go through the protosomal pathway and loaded on the class 1, so that there's not necessarily a strong overlap between what is recognized by CD4 and CD8. In particular, for example, if you look at the flavivirus, dengue, Zika, the targets of CD4 T cells are membrane associated, capsid, for example, but the targets of a CD8 response are not, are more NS3, NS5. But in the case, for example, of SARS-CoV-2, spike is a great target for both CD4 and CD8, and is clearly also a membrane, a surface expressed protein.
Awesome. Thank you so much, Alex. Thank you, Alba. They are the last of our questions for today. So I will thank you both very, very much for those detailed responses, and I'll move into the wrap up.